The oxygen is absorbed in the body via the alveoli of the lungs by diffusion. Then, the oxygen makes its way successively through the pulmonary veins, the heart, the aorta, the arteries, and the arterioles. It is by the arteries that the oxygen is going to be distributed to the tissues. During all its way through the body, the oxygen is carried in the blood in two forms: dissolved in the plasma and erythrocyte water, about 2% of the total, and reversibly bound to hemoglobin, about 98% of the total, because of the low oxygen solubility in the plasma. Oxygen is also stuck in muscles thanks to the myoglobin, an analog molecule of the hemoglobin. Hemoglobin is a tetramer, 4 globin chains in purple and 4 hem groups in green and white. The blood of a normal adult contains at least 6 different species of hemoglobin molecules due to the globin chain, all of which have the same principal structure and function. So each monomer is constituted of one hem, iron atom in green, and one globin chain. The hem group is a prosthetic group, so covalently and permanently bound to the molecule. It is composed of a central iron atom coordinated to a porphyrin ring with 4 nitrogen atoms. There are 6 genes in the human that regulate the globin production, resulting in 6 different polypeptide chains: alpha, beta, gamma, delta, xi, and sigma. Almost all the hemoglobin molecules are constituted of 2 or more different polypeptide chains. For example, hemoglobin A is composed of 2 alpha chains and 2 beta chains, and fetal hemoglobin is composed of 2 alpha chains and 2 gamma chains. When the iron atom in the hem group becomes oxidized, it means that he loses an electron. Its valence state changes from +2 ferrous to +3 ferric. The hemoglobin is then called methemoglobin or ferrihemoglobin, and the ferric iron will not bind oxygen. The level of methemoglobin is maintained at low level by the enzyme NADH methemoglobin reductase. It is important that the level of methemoglobin be kept low because it don't reversibly bind oxygen and thus cannot carry oxygen. Carbon monoxide has a very high affinity for the hemoglobin, 200 to 300 times more than the oxygen in normal adults. The deoxy structure is characterized by the presence of inter- and intra-subunit salt bridge, which give a tense configuration. The oxy conformation is obtained when the salt bonds are broken so as to give the tetramer a relaxed quaternary structure. The iron atom is also covalently bound to the histidine residue F8 of the globin in which the heme is inserted. This residue is called proximal histidine. The proximal histidine is F8 because this is the 8th residue of a F helix, and the distal histidine is E7 because this is the 7th residue of an E helix. The proximal histidine is the closer one to the iron atom, and the distal one is the furthest. The role of the histidine residues is to regulate the ligand binding to hemoglobin in favor of the oxygen.
We are now going to talk about the allosteric effect on the cooperativity in hemoglobin. But first, let's have a general definition of these notions. Allosterism can be defined as the modulation of an enzyme activity via the binding of a receptor to one site affecting the binding properties of a different sites. While cooperativity is the interaction process by which the binding of a substrate to one site on the molecule influences the binding at a different site and cause conformational change in the enzyme structure, which affect the enzyme affinity for substrates. The hemoglobin molecule has 4 distinct binding sites for oxygen, the iron atoms in the 4-heme group. Thus, each hemoglobin tetramer can carry up to 4 oxygen molecules. So here, you can see encapsulated heme group. At this state, the hemoglobin has a very low affinity for oxygen since there is no oxygen attached to it. And it's referred to the thin state, also called the oxyhemoglobin. When the first oxygen molecule binds the iron, it triggers a conformational change in the structure of heme and has a domino effect through the rest of the molecule. The quaternary structure then changes, which increases the affinity for a second oxygen molecule to bind. Moreover, when this second oxygen is bound, the structure relaxes again and has a greater affinity than before for a third oxygen. We are at 75% of saturation, and the hemoglobin has an even better affinity to welcome a fourth oxygen molecule to bind. Now that our molecule is completely saturated, it is at its relaxed state or oxyhemoglobin. Such behavior indicates that a cooperative interaction exists among oxygen binding sites and the hemoglobin molecule. The cooperative binding of oxygen by hemoglobin is one example of what is referred to as an allosteric effect. Allosteric effect in hemoglobin occur when the binding properties of a molecule change as a consequence of a second ligand binding to the molecule, and this alters its affinity toward the first ligand. If the 2 ligands are the same, for example, 2 oxygen in the cooperative binding that we explained before, it is called a homotropic allosteric effect. In the contrary, if they are different, it is considered as a heterotopic allosteric effect. Several factors can influence the binding of oxygen to hemoglobin. First, we have 2,3-bisphosphoglycerate or BPG, one of many allosteric heterotopic regulators. The BPG is a glycolytic intermediate produced in the red blood cell. This molecule binds to the central cavity of the oxyhemoglobin and stabilizes it. The increased stability of the T state makes the hemoglobin less likely to bind oxygen. Let's take now a look to the following graph. The y-axis represents the saturation of hemoglobin in percentage, and the x is the partial pressure of oxygen in millimeter of mercury in our tissues. By looking at the 2 curves, we can see that the binding of BPG to the molecule reduces its affinity for oxygen. Temperature is another allosteric regulator of hemoglobin. Since the tissue cells have a high metabolic rate, they will require a higher supply of oxygen to maintain a proper concentration of ATP. And the only way the cell receives a higher supply of oxygen is if the hemoglobin allows them. Therefore, at higher temperatures in the blood plasma, the hemoglobin will be less likely to bind to oxygen and be more likely to release it in the tissues. The graph shows the effect of higher temperature on the oxygen affinity of normal hemoglobin. Other regulators such as the pH or the partial pressure of carbon dioxide can also affect the binding of oxygen to hemoglobin molecule. As a conclusion, let's briefly present a common disease that affects hemoglobin functionalities: sickle cell anemia. Sickle cell anemia is a genetic blood disease due to the presence of an abnormal form of hemoglobin, hemoglobin S. It is caused by the substitution of valine instead of glutamate at position 6 in the amino acid sequence of the beta-globin chain of hemoglobin. This mutated form of hemoglobin tends to aggregate after unloading oxygen, which causes the red blood cells to change shape. Instead of being flexible and disc-shaped, they are more curved. The sickle cell blocks little vessels, depriving the organs of blood and oxygen, which lead to the periodic episode of pain and damage to vital organs.